welcome to the video on Drug Excipient Compatibility Study Importance. In this video, we are going to see what is the reason behind performing the Drug Excipient Compatibility Study, or commonly called as DE study or DEC studies. Whenever the new formulations are developed, that time the formulation is required to be stable. So whatever the raw materials are being used or would be used, those should be compatible with the API or drug. And to know the compatibility of excipient with the drug excipient compatibility studies are performed. DEC studies are performed during the research phase of the product development, and the compatibility between drug excipient or between drug and drug if the combination product is there, and sometimes between excipient to excipient also is performed. Also sometimes the compatibility of drug with some excipients excluding other excipient is performed, and compatibility or incompatibility may be physical or chemical. If there is change in color, change in appearance, that is coming under the category of physical compatibility. So physical compatibility involves the tests like description, appearance, color, melting, odor, or any other change in the description or physical parameters. Then chemical, change in the assay, change in the impurity profile of the API, increase in any impurity or stabilization of the API, change in the form of the API from crystalline to amorphous, change in the hydrate form, solvent form or pseudopolymorph, formation of adduct. So many things are there coming under the chemical stability or compatibility. As I have mentioned, the drug excipient compatibility study is performed in the research phase or during the prototype development of the formulation. So the basic rationale is to select the suitable excipient, compatible excipient, and low risk excipient or the excipient which will not have any risk for the prototype formulation. It is done to have early risk mitigation in hand for early identification of the stability problems, to study the intrinsic stability of the API, because in the DEC studies API as such is also kept so that it becomes the reference for comparing the drug and excipient mixtures or binary samples. Then the DEC study is to identify the stabilizing effects of excipient alone in the mixture or in combination like in the composition, for accelerating the development of stable prototype formulation, for preventing the stability failures, for reducing the time for bioequivalence study or bioavailability study, and to reduce the time to file the formulation to the regulatory authorities. So it is the preliminary requirement for the formulation development, and in the formulation development stages or product development stages, the drug excipient compatibility study comes under the research stage. Then the DEC study is also important in terms of reducing the stability risk for formation of degradant impurities, formation of adduct type of impurities, formation of nitrosamines, and formation of nitrosamine drug substance related impurities. So nowadays nitrosamines and NDSRIs are very critically evaluated by the regulatory authorities. So in DEC studies also you can consider the nitrosamines, NDSRIs, and also you can study the effect of stabilizing excipients for degradation. Then to reduce the risk for physical and chemical stability or incompatibility, to understand the change in the API form or change in the pH in liquid formulations or the formulations which are impacted by the pH or microenvironmental pH. So in those type of formulations, acidic, basic materials can be used, buffering agents can be used to study the compatibility and effect onto the stability. So in the compatibility study, the API is mixed with excipient in certain ratios. I am going to cover the entire topic in upcoming videos, but for now I will cover the reason or the rationale behind making the drug excipient binary mixture in ratio. So drug to excipient ratio selection is very important, and in this slide we will see how these ratios are selected and what are the pre-requirements or prerequisites or information should be in your hand for selecting the drug excipient compatibility ratios. Always in this the drug is considered as 1 and 1 is to 2, 1 is to 3 or 1 is to 10, 1 is to 15, 1 is to 20. The next ratio is for the excipient. Consider you are making a tablet of 100 mg and in that you are going to use 10 mg of the API and you are going to use 70 mg of the diluent. So in as such a ratio the API is 1 and 1 is to 7, that is 10 is to 70 mg is the actual ratio in the formulation. So you can increase that ratio up to 1 is to 10 or 1 is to 15 so that you can understand the effect of diluent on your API stability. In upcoming videos I will cover that topic in detail. Now how to select the drug excipient ratio? What are the parameters to be evaluated? First is maximum daily dose of the API or of the formulation, and based on that tentatively you can calculate the daily intake level of excipients. Then consider the maximum use levels as per the guideline of IID or IIG database or IID database. You can get the information. Then based on to the reference formulation characterization or reverse engineering for the excipients. Then consider the API load in the formulation, whether your formulation is containing API in low, medium or high concentration. Then calculate the actual ratio of the API to excipient in the prototype formulation which we are going to formulate in the future. That means based on to the R&D characterization you can have the tablet weight, and if you are working on to the novel formulation then you can consider the maximum tablet weight for the dose. Excipient function, whether it is diluent, binder, lubricant, disintegrant or suspending agent based on to your formulation. Then which levels you are going to be considered for quality by design time and considering the critical material attributes of the excipient. Consider the grades of the excipients also. So you can use multiple diluents, multiple disintegrants, number of solubilizing agents or disintegrants or lubricants, film coating agents. So whatever the excipients are likely to be used in future, you can put these excipient in the excipient compatibility study. Also you can put different APIs based on to the API form, API supplier or route of synthesis. Then consider your formulation development experience. Whatever the studies you have performed in your experience, that expertise can be utilized. Also consider the other similar formulations of the same API. Now coming to the importance. So the DEC study is to find out the compatible or suitable excipient for the drug substance. That excipient should be physically and chemically compatible to the API, and it should not have much impact onto the API potency and the impurity profile. So based on to the API potency at initial, and as such API, it should be similar without any significant change with the excipient at some storage conditions like 4075, 3075, 3065, 2560 or 228. So how to make the storage conditions, how to make the samples, that I will cover in the upcoming video. Always consider that the API potency or impurity profile should not get hampered. Selection of suitable or compatible grade of the API or suitable form of the excipient or API. That is the another important reason for DEC studies. Then selection of suitable stabilizer for the API. If you know that the API is prone to oxidation, then you can use multiple antioxidants in single or in combination with the other excipient composition, and based on that you can select the stabilizer for your API. You can select the buffering agent, chelating agent. You can also select the acidifier or basifier, and you can have the data before your prototype formulation is ready. So like this, one by one excipient can be selected and excipient can be removed which is incompatible. Select the excipient which is found compatible and do not go ahead with the excipient which is increasing the impurities. Then another important thing is identification of excipient related to increase in the impurity levels. Then to understand compatibility between drug and other drug in the combination formulation. If you want to use the two drugs in the formulation, that time you should also keep the one drug as a drug and another drug as a excipient. So you can make the ratios, front ratios as per the dose, and then you can understand is there any physical chemical incompatibility between the two drugs. Then selection of the API form, API source and ROS. Suppose you are using amorphous form of API and you want to select the source, or you are using the different crystalline form of the API and you want to select one out of those, then compatibility study can also be done. So you can select the best API form, best API source and the route of synthesis. So based on the results of the compatibility study, you can have the understanding about the intrinsic stability of the API, intrinsic nature of the API and effect of the excipients onto the stability of API. Based on that, you can conclude the future problems or risk in the stability. Many of the times, the DEC studies are performed more than 3-4 times to have complete understanding about the excipients. There should be a clear understanding about the excipient compatibility and stabilization by the excipients. It is not only the evaluation of compatibility, but it is the evaluation of the stabilizing effect of the API also. So the in the upcoming next videos, I am going to cover the topic in detail, but for now I have only covered the reason for DEC study, or you can say importance of the drug excipient compatibility study in the formulation development. So thank you for watching the video.